This is Unit 6B, Part 2. We left off here last time. This is Unit 5B, Part 2. We were talking about antibodies. Antibody molecules, I remind you, are found on the surface of B cells. Each unique clone, or each individual unique B cell that gets produced, has a unique type of antibody molecule on its surface. It'll be specific for one particular foreign target antigen. When I say specific, it means it's able to bind it specifically. It's no different from any other receptor-ligand interaction. You know, the red blood cell growth factor called erythropoietin, which is produced in the kidney and triggers red blood cell production in the bone marrow. Erythropoietin binds to erythropoietin receptors. Insulin binds to insulin receptors. So in this case, a single type of foreign molecule, an antigen that's recognized as non-self, can be bound by the antibody molecules all over the surface of the B cell. Now, activation of a particular B cell occurs when it binds to its target antigen. And with activation, there are two key things that happen. One is it starts to, just give me a second here. I've lost, oh, there it is. No. Two key things happen. It starts to proliferate. In other words, it starts to multiply. So the cells divide into two daughter cells, and then the two daughter cells, etc. And each division of a B cell would be roughly, say, 12 or 14 hours, 16 hours at the most. And sure, you start with only a very tiny number of cells that were activated, but as it doubles with time, it gets to a huge number of cells. So you're getting more and more and more cells with a particular type of antibody on their surface present, because they're proliferating, they're multiplying. The other thing is that they start to secrete the antibody molecules. The antibody molecules are no longer just on the surface of the cell, but they're actually being secreted as soluble antibody molecules that can go out and go throughout the body and the plasma, and sometimes they're secreted out onto mucosal epithelial surfaces. It depends on, only certain types can be secreted. But the antibody molecule goes all through the body, okay? And it's still, even in a soluble form, capable of binding specifically with that foreign antigen molecule that initially activated that first B cell that had a specific antibody for the antigen. So you end up with lots of B cells, all the same to each other, so they're all a single clone, they're clones of each other, and they're all secreting this one type of antibody molecule. This is a diagram of an antibody molecule. It's an artist's interpretation of the antibody molecule. This is a different kind of representation, which is computer generated. Suffice it to say, what you have are 4 polypeptide chains. So 4 protein chains, 2 smaller ones, which are called light chains. That's what the L stands for, light. And 2 longer, also identical to each other, just like the light chains are identical to each other, 2 longer polypeptide chains that are called the heavy chains. They're held together by disulfide bonding, and the disulfide bonds are shown here. There's these intra-chain disulfide bonds, but there's also inter-chain, between chains. So the heavy chains are held together, the light chain is held to the heavy chain. It becomes one single soluble molecule. Now, what else can I tell you? I can't go back, unfortunately. I forgot that part. Just a second. Yeah, I can't go back. There was something I wanted to tell you in that previous slide, but let's move on. I think it'll come up again. So this is a different kind of representation. It's what's called a ribbon diagram. This is a standard type of biochemical representation of a molecule. It's a protein. Remember, the antibody molecules are proteins. The two light chains are shown in red. They're identical to each other. The two identical heavy chains are shown in blue and yellow. And this kind of ribbon part folding back and forth on each other, you might recall that you learned, I believe, that proteins have secondary structure, the beta pleated sheets. So these areas here, this area here, here, let's try this color. This area here is a beta pleated sheet. There's a beta pleated sheet. There's one on the light chain. There's another one on the light chain, etc. Now, it's important for you guys to realize that when you look at this molecule, it is not static. It can move around in space. It's a three-dimensional object. And in here, this area here is very, very flexible. So these are like two arms that are waving around. These two areas here and here, they're like two arms that are waving around. Here we go, back to that. Similar, not exactly the same as what I had before. Again, we have two identical light chains. Right? There's one and there's the other one. And two identical heavy chains, there and there, held together by disulfide bonding. And what you should understand is that the part of an antibody molecule that is the receptor for a foreign target antigen is in this area here, in this area up here. Okay, so this area between the light and the heavy chain is very specific for a particular molecule. It can bind to a particular foreign molecule, an antigen. And here on this one is an identical binding site for the same antigen. Let's call it, for now at least, antigen X. Okay, so it can bind to antigen X. Now, if you looked at a completely different B cell that was produced out of the bone marrow, it would have similar antibody molecule, the ability to produce similar antibody molecules, but the antigen it bound to, the antibody it produces would bind to a different, would be specific for a different antigen. So each antibody molecule can bind to a particular antigen, and the binding sites are at the ends of these two arms made up of the light chain and the heavy chain. Let's get rid of some of this drawing that I've done here so we can see things a little clearer. Okay. Now I think what I'd like you to look at here is the fact that from one antibody molecule to another, produced by totally different B cells out of the bone marrow, you will have these two heavy chains, and they have domains, what are called areas or domains in them, that are constant. Okay, every single antibody molecule produced by every single B cell will have in it these constant regions indicated by a C. It's only up here in the antigen binding site that they will be different from each other, only up here. But otherwise, they do have constant areas that are the same. And those constant areas are constant heavy 1, these what I call domains or regions. Constant heavy 1, that's this part here. Constant heavy 2, that's this part here. And constant heavy 3, this portion of the polypeptide or protein strand. Similarly, the other one, remember they're identical to each other, has a constant heavy 1, constant heavy 2, and constant heavy 3 domain. At the very end, you have the variable heavy domain and this variable heavy domain. This is the heavy chain all along here with 4 domains. A variable heavy, constant heavy 1, constant heavy 2, and constant heavy 3. And the same for the other strand. Similarly, not identical, but similarly, in a similar way, we have for the light chain, we have a variable light domain and a constant light domain. And over here, this one's identical, variable light, constant light. This is the antigen-specific binding site. It's specific for a particular antigen. And it's made up of the variable light and the variable heavy domains. Similarly here, again, the same antigen if we're on the same molecule. Okay. Let's move on. Oh, there's that diagram that I showed you earlier. So 2 light chains, 2 heavy chains. The heavy chains have variable heavy, constant heavy 1, constant heavy 2, and constant heavy 3. And all of that's written out here. Here, the heavy chain is made of these domains, light chain is made of these domains, and the antigen-specific binding site are the two. There are 2 antigen-specific binding sites at the tip here, 1 and 2. Okay. Remember, I told you that in this area of the molecule, it is very flexible, so that these 2 arms can wave around. They're not always sitting there at this 90-degree angle to each other. You see that's 90 degrees. This is a very, very flexible structure, so it can wave around and is able to bind with a really, what's called an affinity, a really high affinity. A surprisingly high affinity for its specific target antigen. Affinity. Not that easy to write or spell with these things. Okay. So antibody molecules that bind to different antigens each have different variable regions. You know, if you look at one particular B cell that comes out of the bone marrow, it has antibody molecules on its surface specific for a particular antigen. And another B cell will have antibody molecules specific for a different antigen because they have slightly different or different variable regions in the light and the heavy chain from each other. These 2 are the same, they're on the same molecule, but otherwise they're very different from the next cell, what the next cell can produce. So they form this kind of Y-shaped molecule. It's very flexible. The reason it's flexible and the area that allows it to be flexible is called the hinge region. Hinge, like hinge on a door. The rest of it, the constant areas determine the class of the antibody. Antibody molecules are separated up into different major classes, and they have common functions. You know, there's one major class called IgG, and all IgG molecules, all IgG antibody molecules, doesn't matter which clone produced it, if it's an IgG, they all have certain common functions that they can mediate if they bind up here to their specific target. Once they bind to their target, then they will be able to carry out the functions that the rest of the molecule, the constant areas, allow them to do. So the C regions are constant areas that are forming this stem here of the Y, not the arms, but the stem. This is the stem of it. They dictate which cells this will bind to. Down here it can bind onto the surface of certain types of cells, and it also determines the class and how it acts, functions biologically. Up here is a binding area. Okay, now please don't get clonal deletion and clonal selection mixed up. I told you that if a B cell is produced in the bone marrow and it has antigen binding regions or at the end of the arm, if the antigen binding sites will react to self, then it undergoes clonal deletion. It gets killed off, destroyed. Otherwise, it would react to your own tissues when it got out of the bone marrow. Same with T cells when they mature. Anything that's autoreactive gets deleted. That's called clonal deletion. No, you don't want that kind of clone in you. There's another process which is called clonal selection. I'm going to describe it here for B cells, but it also operates the same idea with T cells. So in terms of what it means in terms of clonal selection is that each, as you know, hopefully by now, and understood, that each individual unique B cell that gets produced out of the primary lymphoid organ, the bone marrow, has a particular unique specificity for a certain target due to the variable regions at the end of the two arms. And if it binds to its target antigen, if it binds to the target antigen, it gets activated, starts to proliferate. The cells replicate, they divide and divide, and starts producing antibody. So each unique B cell and any of its daughter cells together form a single clone. They're all identical to each other. And the receptor, of course, for the antigen is at the ends of the antibody molecule. So if you produce out of your bone marrow 1 billion different B cells, they will have 1 billion different antibody molecules, each able to bind to a different antigen molecule. You produce B cells, a tiny number that can, if you look at all of them, they pretty much produce, there's a B cell for almost anything that could get into you. If you're looking at a single mature B cell, it'll recognize its target via the ends of the antibody molecules on the surface. These are the antigen receptors, the antibody molecules, the two antigen binding sites at the ends made by the variable light and variable heavy chains. If it contacts and binds its antigen, it becomes activated or stimulated, it starts to proliferate. In other words, stimulates the B cell clone to proliferate, multiply, and the cells will do two things. The new daughter cells will go on to form two different types of cells. Most of them will form into plasma cells. A plasma cell is a B cell that's been activated and gets bigger and can produce a lot of antibody protein. AB stands for antibody. So it can produce really incredible amounts of antibody. So it starts to be a protein factory putting out lots of antibody. They're called plasma cells. But some, a reasonable number, some of the B cells, of the ones that, the daughter cells from that clone that was activated, some of them become memory cells. So they don't go towards the plasma cell and start producing lots of antibody. Instead, they become memory cells, and these are long, very long-lived cells that could respond if you get exposed much later down the road to the same foreign molecule. And this point here, this production of memory cells, is why if you get exposed to something, you become immune. The next time you will be able to respond really quickly and strongly, and if it's an infectious disease organism, you will not get infected, because your memory, you have way more cells now. You have these memory cells sitting there, and they'll get activated and they'll multiply, and they'll quickly kill off or destroy that foreign organism. This is why you don't get infected again with the same thing. This is why vaccination works. Because with vaccination, you're getting exposed to a safe form of an antigen associated with a microorganism, and you're generating memory cells. So the next time, if you get infected by the organism, your memory cells now, instead of just a very tiny number of B cells that have the right receptor, you have lots of cells. Okay. Now, what this means is that out of all the different unique clones B cells, the individual different B cells that you produce out of your bone marrow, there's going to be a tiny number that can respond to a particular foreign molecule, and essentially you select the right clone. That's called clonal selection. You get exposed to a particular antigen, the right B cell with the right receptor will be activated, proliferate, secrete antibody. So that's a process of clonal selection. That's how your antibody producing B cell defense works. Remember, we're talking about the adaptive immune response. They call it the adaptive immune response because it's not ready immediately. It has to adapt first to the presence of its target antigen and proliferate, and then start multiplying, and then start secreting antibody. So you get a particular foreign antigen in you because you get infected with a microorganism, and you have a few B cells that can respond. Okay, here's one that can respond. You have another 100 million, 200 million, even more, probably even a billion B cells in you, but they have the wrong receptor. Only this one can respond. So this is the one that gets activated. This is the one that divides, and then divides again and again and again. Now, if you look at a cell and you have two daughter cells, and it divides, say, every 12 hours. So, okay, after one day, 12 hours, you go from 1 to 2, and then you go to 4. But after, you can sit down and write this out if you want, that's 12 hours here, and another 12 hours here, so you go to 4. But if you go all the way out 12, 14 days, you have a huge number of cells being produced. This one was selected, clonal selection. The right clone was selected by the presence of the antigen that it can respond to, produced many of these daughter cells, and then most of those become plasma cells that can secrete that antibody that's specific for that antigen. But a few become memory cells and sit and wait. So this is an initial primary response if you've never been in contact with the antigen. But here later, if you have memory because you were exposed before, then you get a secondary response, and the secondary response is much quicker, faster, and protects you. Now, in order to have hundreds of millions of unique B cells in you, each with a unique antibody on their surface, you have to have a lot of genes in, you know, each one coded for, they're proteins, so they're coded for by genes. You know, if you look at the human genome itself, it's not big enough to contain enough genes to code for all the possible antibody molecules that all the different B cells can produce. It just doesn't contain enough genetic information. So how is all of that, how does all of that come about? I mean, if there's not enough DNA in the nucleus of a cell to code for that, and the cell's going to become a B cell, how can it do that? It occurs because there are multiple choices. Genes for antibody molecules, at least for the two types of polypeptide chains, the light chain and the heavy chain, are present as gene segments. So there's several different gene segments coding for each of those proteins. There's a number of different gene segments, and each gene segment has many, in the DNA of the nucleus, there are many possible gene segments that could be used to build up and recombine together to build a gene coding for the whole strand. So, but this is a random process how which gene is chosen, which segment is chosen. They get shuffled, well, shuffled's not the right word, but they get recombined into a newly assembled gene, and that will express or code for a particular specific antibody molecule. I don't know if the next. Okay, I'm not sure where I was exactly. I was interrupted by a phone call. So, you know, you have different types of antibody molecules in terms of classes. 5 classes, G, M, A, D, and E. The antigen binding sites are still, you know, here on IgG or IgE. Nobody talks much about IgD. Not quite clear what the function is, probably. I don't think it's been worked out that well, actually. So IgE and IgG have the usual 2 antigen binding sites. You know, these molecules can be represented in many different ways, and these are just artist drawings of the molecules. Again, this is a dimer, IgA, so 1, 2 binding sites, 3, 4 actually, because it has it's a dimer, it has 2 of them, and 1, 2, 3, 4, 5 on this pentamer, IgM, so it has 10 antigen binding sites, all directed at the same antigen, but 10 of them. Let's move on. Now, I talked to you earlier about primary and secondary response when a foreign antigen enters into the body tissues. A foreign molecule could be anything, including molecules on a dare I say, virus or bacterium. When it enters the first time, because of the process of clonal selection, if it establishes and starts to multiply, if the microorganism starts to multiply in the body, then molecules from it will at some point contact a B cell that has the right antibody on its surface that can react to it. And if that's the first time, then that will trigger a primary response. So the very first exposure is a primary response. And the type of antibody that gets produced is always going to be a IgM class. Okay, so you'll get that IgM kind of pentamer type of response. And then later, it switches over to IgG production. So the initial exposure, this is time along the horizontal axis, and this is antibody titer. This means just exactly how much antibody is present in the plasma. They would be in the serum. They would be testing the serum. So what you have here is initial exposure. The IgM is shown by the red line. IgM production is shown by the red line. IgG is by the black line here. So the initial production is always IgM, and that rises up, and then it's followed by an IgG class antibody. Same antigen specificity. The target that the antibody can bind to is the same, not any different, but there's a switch over. It's actually called the class switch. So the IgM class will be switched over to the IgG, and it'll start producing IgG antibodies. Now at some point, and because it's a graph and they have to show you some example here, it's just an example. They're showing the secondary exposure to antigen here at I don't know what this is, maybe 24 days. So they picked a spot. I mean, it could have been at 124 days. It could have been at 505. It could have been 2,000 days later. But the second exposure, don't forget that after a primary response where you get plasma cells proliferation with binding, you get activation, proliferation, secretion of antibody. But some of the cells, the daughter cells that get produced, go to memory. And the antibody titer, actually the amount of antibody in the serum drops off with some time. And then, but upon secondary exposure, because you have all those memory cells, the response is much faster. And then with secondary response, the secondary response is mostly IgG rather than IgM class. Here's another example. In this case, they gave the secondary exposure as 28 days, but here, the initial antibody response to antigen X, they're calling it X, is here, and the secondary exposure is here. Okay, much bigger. Now, if you gave not only antigen X for the second time, but a completely different antigen at the same time, and it was the first exposure, then the antibodies that the body will produce will be just like that, a primary response, just like here, no different. So we talked about clonal selection of B cells, and the same kind of idea occurs with T cells. There is clonal selection of T cells. The bone marrow produces the T cells, precursors, primitive T cells that are going to become T cells. They migrate through the bloodstream to the thymus, where they mature, become immunocompetent, and the ones that are self-reactive, autoreactive, they get deleted by clonal deletion. But the ones that are not self-reactive, that have receptors for foreign molecules, they will exit and populate the body, the bloodstream, the different secondary lymphoid organs. Each cell that emerges out of the thymus is a clone, a unique clone that has receptors targeting a single foreign molecule. All right. Their receptor is a T cell receptor, and it's all over the surface, but it's reacted to a single type of antigen target. The next cell would be comes out would be to some other antigen. So T cells recognize they're able to bind their target antigen via the T cell receptor on the surface. These are receptors for that antigen. And that interaction with the antigen activates the T cell clone. It stimulates the cell with the that had that receptor for the antigen to start multiplying. And the cells multiply and form one of two major types of T cells, either a T helper cell or a T killer cell. And T helper cells, you might see them sometimes referred to or written as T H, like that, T helper cell. And T killer cells, you might see them written sometimes as TK cells. Occasionally people use this term cytotoxic, so TC. There is no difference. These are the same. They're TC, TK cells are the same. Okay, I didn't say a lot about T cells, but T cells actually are the major way that the body immune system, the adaptive immune system, protects us against virus infections. Virally infected cells will express on their surface proteins that are viral. You will get viral proteins on the surface, and T cells, T killer cells, can, with the right receptor, and somewhere in your body you'll have a T killer cell that has the right receptor for that viral antigen, will bind and target and kill the infected cell. By killing a virally infected cell, you stop new virus particles from being produced. So your immune system defends you by killing off the infected cells. Okay, let's talk now about something different, a more general topic about immunity and acquired immunity. We acquire immunity by being not in a number of different ways. Now, and there are 4 different types of acquired immunity. We are able to gain or acquire immunity during our lifetime by exposure to different foreign molecules. And that can happen in a number of different ways. Now, immunity can be acquired naturally, and it may be active immunity. And this is the kind of immunity that is the result of an infection. You get infected with a particular microorganism, a bacteria or a virus, and your immune system responds. And if the innate immune system fights it off, then you're fine. But if it can't immediately fight it off and the infection persists longer, then the adaptive immune response will be triggered, and you will get T cells or B cells being activated, the ones with the right receptor, and proliferating, and the B cells will produce antibody and the T cells will become T killer cells, and you become immune. And both types actually can form memory cells. And so you are immune. You have acquired immunity. You did it naturally because it was the result of an infection, a natural process. But it was an active response through clonal selection. That's an active process. The cells with the right receptor for that, let's say it was a virus, cells with the right receptor were activated, proliferated. All that's a natural active immune process. So naturally acquired active immunity. There's also what is called naturally acquired passive immunity. It's passive because the individual's immune system is not what's mediating the response. They're passively receiving that immunity because you can get transfer of antibody molecules from the mother to a developing fetus across the placenta. All right, so this is a transplacental passive acquired immunity. So natural, yes, of course, across the placenta, it is a natural process, but it's passive. The fetus's lymphocytes, either T or B, weren't activated. It just got the antibody molecule that the mother was producing. And those antibody molecules they're just proteins. They don't stay forever It's not like you have a naturally acquired active response where you have memory cells and the next time you get infected with the same thing, there are many more, so they proliferate. No. It's passive. You just get the protein. It's not going to hang around forever. The other thing is, of course, the baby is born and it doesn't receive any more antibody across the placenta, obviously, but babies will receive antibody via colostrum. Colostrum is the early milk that is produced by a pregnant woman. So, you know, if you're talking about breastfeeding versus formula, you can't beat hundreds of thousands, if not millions of years of evolution that allowed antibody molecules to be in mother's milk, in the early mother's milk, colostrum, which is called colostrum. And that way it gets transferred into the baby and protects the baby. But that's, again, yes, natural, but passive. It's not the baby's immune system that's providing the immunity, it's the mother's that's getting transferred to the baby. Let's talk about another two types. There are two other types of acquired immunity. They're both artificial because they involve an injection, and that's not a natural process. And one is active and one is passive. Now remember that active processes involve activation of the individual's immune system. And in this case, that's what's happening when you get vaccinated or immunized. You actually get an injection of the antigen that's associated with a microorganism. You get injected with some protein molecule from that's found on the surface of a bacteria, or a protein that's found in association in the protein coat of a virus. And by injecting that in, your immune system responds actively and you acquire immunity. So you have artificially acquired active immunity with vaccination. Sometimes, however, people get infected or exposed, and with exposure we assume infection, with something that is so deadly that there is no time for their immune system to respond. They'll die if they have that. If you get bitten by a rabid dog who carries a rabies virus, there is no time for your immune system to protect you. You'll be dead soon, well before your immune system can respond. And so what they inject you with is antibody that is either, well, preferably produced in a lab as a monoclonal antibody. It's artificially produced in the lab. But if not, then they try to get antibody from people who had been exposed to and were now immune. So that's an artificial system because it's injected, but it's passive. There's no activation of your immune system itself. It's just giving you the antibody that'll protect you. An antibody specific for example, for rabies. The same is done for snake venom. If you get bitten by a snake, by the time your immune system kicked in 10 to 14 days later, you'd be long dead. It is so toxic, and so they give you immune serum from someone who's been exposed. Antibody, in other words. So that's artificially acquired passive immunity. And the last slide here reviews that and presents it in the same thing. There's natural, there's artificial acquired, there's active and passive, natural, active and passive, artificial. And that's the end of Unit 6B, Part 2.